wait for 5 minutes to let others join, and then we can start. Good evening, everyone. We'll be starting now. Hello and welcome to week 6 of the PMRF-NPTEL session on animal physiology. Today we are going to cover the lectures titled Neural System 3, diving even deeper into the neural networks, how they exactly work, and how exactly spatiotemporal summation of neural networks work, and how is memory stored in a particular system such as ours. So I will briefly introduce myself. My name is Anupam Chatterjee. I am a PMRF fellow and PhD scholar at IIT Kanpur, and I will be hosting these sessions on Tuesdays. So how exactly we do this is that we will be solving the assignment questions from the previous year's run of the animal physiology course and go through each of the assignment questions and try and answer them, and in that way better understand the overall topics that are taught throughout the week and better know how exactly to solve the final assignment questions. So does anyone have any questions from this week's lectures on NPTEL? Then we can address them first and then move on to the assignment questions. Okay, I think no one has any questions from this week's assignment, this week's lecture series. So we'll be moving on to the assignment questions. So this is the playlist or YouTube playlist onto which all the recordings of these live sessions are uploaded. So if you have you are unable to actually attend the live sessions or want to go through one of the parts of the live sessions again, then you can just log into YouTube, go to the playlist and watch it yourself, and then solve any doubts you had. So this is the first question that we will be tackling today. So how exactly we do this is that we will be you will be shown an assignment question, and I will ask you to put into whatever you think is the correct answer into the chat. So A, B, C, D. Then we'll be discussing the question and answering it. So the first question for today is, the longest process of a neuron is called dendrites, dendrimers, axon or axemer. You can put whatever you think is the correct answer into the chat. Vishwajit says it is 8 dendrites, the longest process of a neuron. Anyone else wants to try and answer this? You can put whatever you think is the correct option into the chat. Anyone else wants to answer this? The longest process of a neuron is called A, B, C, D. 4 options are there. You can put whatever you think is the correct option. Okay, so let us go ahead and discuss. So this is what the anatomy of a normal general neuron looks like. You have a nucleus surrounded by a cell body, and this entire cell body has various cytoplasmic extensions. These extensions are often short and surround the cell body itself or the soma. So these extensions are known as dendrites. But one of the dendrites is often much longer than the other dendrites, and it's pretty long. This could be like 2-3 times the size of the cell body, upwards of say feet in length, and this is known as the axon. So this part is the axon, and these are the dendrites. And axon has axon is then divided further into number of processes at its end, from where signals will be transferred to the next neuron. So how neurons function is that they act as a unidirectional flow of information. They receive information in the form of electrical or chemical signals on the dendrites, so on the left, and the axon is the one that helps in the transfer of information towards the next neuron. So always the information is going from the left to the right in the image, so from the dendritic end to the axonal end. Further, the axon has number of other properties. This includes that the neck region, where the axon actually originates from the soma or the neuronal body, is known as the axon hillock. So we had discussed in the previous session that this axon hillock is filled to the brim with voltage gated sodium channels, which allows the action potential to begin right here. So we will be discussing more about the action potential and how that works a bit in this session as well. Further, the axon is covered with a number of smaller cells. These cells produce a huge layer of lipids which covers part of the axons, and these are the Schwann cells, and this covering is known as the myelin sheath. This helps in conduction of the electrical signals through the axon. And finally, there are spaces left between the Schwann cells where the axon is exposed, and these regions are known as the nodes of Ranvier. So in general, this is what the anatomy of the neuron looks like. So to answer our original question, the longest process of a neuron is called the axon. Anyone has any questions from this? So this is basic information of the neuron that we'll start with today, and then move forward to much more complicated topics. Anyone has any doubts from this? Okay. The next question is the conduction speed in neurons blank with effective myelination. So effective myelination does what to the conduction speed of information through the neurons? The options are: it increases the conduction speed, it decreases the conduction speed, it does not change the conduction speed, and finally it cannot be calculated. So we have Vishwajit who says it is A, increases the conduction speed. Aruna also says it is A, increases the conduction speed. Anyone else wants to try and answer this? You can put whatever you think is the correct answer into the chat. Okay, so we have a video here that I will like would like you all to see on how myelination works. I will be shifting to another. I hope the screen is visible. The video is visible, right? Okay, just let me know if it is audible or not.
In this video, I want to talk about the effects of axon diameter and myelination because it turns.
Is it audible? Okay.
Turns out that larger diameter axons conduct action potentials faster than smaller diameter axons, and axons with a myelin sheath on them also conduct action potentials faster. So first let's consider the diameter of an axon and how that affects the speed of action potential conduction. An axon with a larger diameter offers less resistance to the movement of ions down the axon, causing ions to move faster down the axon and causing the action potential to be conducted faster. And let me show you the way I think about this by just considering a single sodium ion that's entering an axon through a voltage-gated sodium channel. So we'll say we just have a single sodium ion in here we're going to look at, even though there's many sodium ions flowing in through these voltage-gated sodium channels. And let's consider this for both the large diameter axon and the small diameter axon. Here's our sodium ion in the large diameter axon. Now both of these sodium ions, once they're inside the axon, could move in really an infinity of directions. They could kind of go in any direction and any degree of direction so that there's an infinity of pathways these ions could travel. Now, if they happen to travel backward in the axon back toward the soma, or if they happen to travel perpendicular to the long axis of the axon, that won't really contribute much to the action potential other than the effects they'll have on the other sodium ions that are coming in. But if they're heading in any of these directions down the length of the axon, that's going to contribute to moving the action potential down the axon. And the same is going to be true for this sodium ion in the small diameter axon. It could go in an infinity of directions as well, just like in the larger diameter axon. But now let's consider the obstacles to this sodium ion moving down the axon. First, there's the membrane of the axon, and then there are all sorts of structures in the cytoplasm of the axon, such as vesicles or large proteins, and then there are filaments and there are tubules, and there are all sorts of structures in the cytoplasm that would pose an obstacle to the movement of this sodium ion. And the concentration of these obstacles would be the same in the smaller diameter axon, but of course there's less cytoplasm, so there would be fewer of these obstacles, but the same number for any given volume of cytoplasm. So if we consider this to represent the obstacles in the way of these sodium ions moving down the axon, what we see is that there are fewer potential pathways for the sodium ion to move down the axon in the smaller diameter axon before it runs into something in a fairly short distance. So let's say this sodium ion heads this way, that gets it a pretty good ways till it collides into the axon membrane. Or if it goes this way, that also gets it a pretty good ways till it collides with the cell membrane. But if it heads this way, it collides into one of these cytoplasmic structures pretty quickly. Or if it heads in any of these directions, it runs into the axon membrane very quickly. Now if we compare that to the sodium ion in the large diameter axon, it has more potential pathways to travel before it collides into something. I mean, it certainly has the probability to collide into things in a pretty short distance, but there's a higher probability that it will travel a farther distance at a faster speed because there are just so many more pathways that it can take to make it a farther distance before it collides into something. So that for each individual sodium ion, the probability is higher that it will travel faster over a longer distance than the probability of the sodium ion in this smaller diameter axon. And then when you consider that many sodium ions are coming in and you average all those average speeds together, together, it's going to turn out that in the larger diameter axon, the ions are going to move faster on average down the axon because they have more potential pathways to travel before colliding into things. And since the speed of action potential conduction is related to the average speed of ions moving down the length of the axon, then action potentials will be conducted faster down a larger diameter axon than down a smaller diameter axon. At least this is the way I like to think about it. But now let's consider the other thing that really speeds up the conduction of action potentials down axons, which is the presence of a myelin sheath around the axon. The speed of action potential conduction is faster in myelinated axons, like I've drawn here with the myelin sheath in yellow, because the capacitance of the membrane is reduced in the myelinated segments, which decreases the number of ions and the time needed to change the membrane potential in these areas. In this context, the word capacitance refers to the number of ions that can be stored in the layers on both sides of the membrane at any given membrane potential, because the membrane potential reflects the strength of the charge separation for any particular charge carrier, like a single cation or a single anion, but the total number of charges along the membrane is the capacitance of the membrane, a word derived from the same word as capacity, or the amount of charge that can be stored. One of the principles of a capacitor, like the cell membrane, is that the closer the charges are to each other, the more charges can be stored on both sides of the capacitor. The way I like to think about that for this is that at the nodes of Ranvier, there's only the normal thickness of the cell membrane, which is relatively thin, so that an anion in the layer against the inside of the membrane is strongly attracted to a cation in the layer on the outside of the membrane. Because they're very close to each other, they're attracting each other very strongly. And because this attraction is so strong, it's able to overcome the repulsion that the like charges feel to each other on either side of the membrane. So all these positive charges, these cations, repel each other because like charges repel, but with this very small distance between the opposite charges on the other side of the membrane, this strong attraction overcomes the repulsion that the like charges are feeling on either side of the membrane. So that on the thin membrane at the nodes of Ranvier, you can pack in lots of charges. You can pack lots of anions on the layer along the inside and cations in the layer along the outside, at least at the resting potential, when it's more negative inside than outside. But the myelin sheath you can think of really as making the membrane much, much thicker, because the myelin sheath is just membrane that's wrapped around the neuron membrane many, many, many times. So now you have a really thick membrane. So in terms of a capacitor, the distance is now much greater between the opposite charged ions on either side of the myelin sheath membrane. So while they're still attracting each other, so there'll be a cation out here and an anion right here, and they are still attracting each other, they're trying to pull toward each other across the membrane that won't let them pass. But the strength of this attraction is much less, because the distance is much farther between these ions, so that now you can put less like charges on the same side of the membrane from each other, because there's less of an attraction across the membrane to make up for the repulsion that these like charges are feeling toward each other. They want to move as far away from each other as they can, and so they're spread out much farther. You can store less charges or less ions along both sides of the membrane because the distance between the opposite charges is farther away. So in a myelinated axon like this, there'll be this alternation. At the nodes of Ranvier, which is high capacitance, there'll be lots of charges, and in the myelinated segments, which is low capacitance, there'll be less charges. For unmyelinated axons, they're basically all just like the nodes of Ranvier. They're high capacitance, so they store lots of charges on both sides of the membrane. Now consider when the action potential starts and this voltage-gated sodium channel opens. Sodium will flow into the axon, bringing positive charges inside. So this area of the membrane is going to depolarize. And to do so, some of these negative charges are going to need to leave the inside of the membrane and mix in with the rest of the ions in the cytoplasm. And on the outside, some of these charges will have to leave and mix in with the ions in the interstitial fluid. And this will take a little time. And it'll take more time here on the high capacitance membrane at the nodes of Ranvier, or the trigger zone, than it will here in the myelinated segments, just because there are less charges that have to move off the membrane to change the membrane potential. So because fewer ions and less time is needed to discharge or depolarize the membrane in the myelinated segments, which has a lower capacitance, or to recharge the membrane or repolarize the membrane when potassium starts flowing out during the falling phase of the action potential, the action potential is able to travel faster through these myelinated segments than it can through the nodes of Ranvier, which is why we see slower conduction of the action potential through the nodes of Ranvier than through the myelinated segments, which we call saltatory conduction, where it looks like the action potential is jumping from node to node. Myelination also decreases the membrane permeability to ions, so that fewer ions in total cross the membrane during an action potential. Therefore, fewer ions in total need to recross the membrane after the action potential through the sodium-potassium pump. That's going to be pumping back out all the sodium ions that came in through the voltage-gated sodium channels and pumping back in all the potassium ions that left during the action potential. And since this process uses energy, myelination increases the efficiency of action potential conduction in terms of the energy needed to maintain these ion concentrations after action potentials. Myelinated axons have most of their voltage-gated ion channels at the nodes of Ranvier, so that while an action potential is conducted faster through a myelinated segment, it actually does decrease in size a little bit as it's going along. The nodes of Ranvier, therefore, are necessary to regenerate the full size of the action potential so that it can continue all the way down the axon.
I hope that was easily digestible and easy to understand why exactly we have the myelin sheath and how exactly it increases the velocity of uh, information travel via a neuron. If anyone has any questions from the video, you can please ask and we can talk about it. Okay, so yeah, coming back to the question, the conduction speed in neurons increases with effective myelination. Any questions from this? Okay, moving forward, the next question. Schwann cells are option A, myelinating cells in the CNS or the central nervous system, myelinating cells in the PNS or the peripheral nervous system, myelinating cells in CNS and PNS, and myelinating cells specialized only for nerves in the stomach and not found anywhere else. So you have four options A, B, C, D. You can put into the chat whatever you think is the correct option. Put whatever you think is the correct option to the chat so we can move forward. Okay, I guess we do not really have an idea about this, so we'll be talking about it. So the Schwann cell, as I have said, are these specialized cells that cover the entirety of the axon present in the neuron and produce this thick sheath of lipids, which is known as the myelin sheath. We have just seen that the myelin sheath actually helps in the saltatory the saltatory conduction of the electricity down the entirety of the axonal terminal, and this essentially increases the conduction speed of the axon. So how exactly do they look like? They'll, in a anatomical view, this looks like something like this. So here we have the middle channel, which is actually the axon, and covering it in different layers is the myelin sheath, and here is the nucleus of the Schwann cell. One important thing about the Schwann cell is that they are only present in neurons in the peripheral nervous system, not the central nervous system. So this was about the Schwann cells. So coming back to the question, Schwann cells are myelinating cells present in the PNS. Moving forward, the opposite question, oligodendrocytes are blank. Myelinating cells in the CNS, myelinating cells in the PNS, myelinating cells in CNS and PNS, and electrically active non-myelinating cells in both CNS and PNS. So again, four options A, B, C, D. You can put whatever you think is the correct option into the chat. Dibya says it is A, myelinating cells in the CNS. Anyone else? Okay. Sibani also says it is A, myelinating cells in the CNS. Okay, so as we know that there is a very distinct divide between the central nervous system and the rest of the body. This divide is known as the blood-brain barrier, which prevents any interaction of any cells or anything else to leach into the central nervous system or move into the central nervous system. This is because central nervous system is essentially very well protected by the body, so that nothing could interfere with its function. So there are some specialities and differences between the central nervous system and the peripheral nervous system. One of the important ones is that there are these glial cells, which are non-neuronal cells that take care of essentially everything in the central nervous system. These include astrocytes, microglia, as well as oligodendrocytes. The oligodendrocytes take the place of Schwann cells in the central nervous system and are these big cells, as you can see right here, which send out cytoplasmic projections that actually cover the entirety of the axon in central nervous system. So you can see the oligodendrocyte as a big cell which sends out cytoplasmic extensions and covers different neurons at once. So this one cell can essentially cover a lot of different neurons. Whereas the Schwann cells in the peripheral nervous system are individual cells that sit on one axon of one neuron, and multiple Schwann cells are required to cover the entire axon. So here is a better magnified view. You can see nucleus of the Schwann cell and the entirety of the Schwann cell covering only one axon, and you need multiple of these. Whereas oligodendrocytes have bigger cells which are present away from the neuron, but has cytoplasmic extension that cover the axons of multiple neurons. So here is a bit more in-depth difference between the Schwann cell and the oligodendrocyte. Schwann cell is a glial cell that is wrapped around the axon of a nerve cell in the peripheral nervous system, whereas oligodendrocyte is a glial cell which has several slender processes which wraps axons of the nerve cells in the central nervous system. So one is PNS, one is CNS. Schwann cell is derived from neural crest cells. So this neural crest are a particular cell type that are developed during or that are produced during early development, and these are cells which are released from the folding neural tube, and these cells can travel a long distances, essentially migrate long distances and form various structures, and one of these include Schwann cells. Whereas oligodendrocytes have a specific progenitor which is known as the oligodendrocyte precursor cell. These insulate cells in the PNS, these insulate nerve cells in the CNS. Oligodendrocytes are capable of only insulating one single axon, and you need multiple of them. An oligodendrocyte can actually insulate 50 different axons at once. Schwann cells can be myelinated or non-myelinated, whereas oligodendrocytes are always myelinated. So coming back to our question, oligodendrocytes are myelinating cells in the CNS of the central nervous system. Any questions from this? Okay, so moving on to the next question. The signal travels within the neuron blank and between two neurons at the synapse via blank. The options are electrically, chemically, electrically, or chemically. So inside the neuron, what type of transmission is this? And outside of the neuron, that is at the synapse, what type of transmission do we observe between neurons? So A, B, C, D, there are four options. You can put into the chat whatever you think is the correct one. Achal says it is B. Sivani also says it is B, that the signal travels within the neuron electrically and between two neurons chemically. Okay, anyone else wants to try and answer this? Okay, so yeah, so we'll be discussing a bit, brushing up on our already acquired knowledge of action potentials and how exactly the electric signal travels within the neuron. So as we know that the information within the neuron travels electrically. So how exactly does that happen? That happens through three different ion channels present on the neuron's body. So a neuron is an electrically excitable cell type. That is, it responds to electrical signals, and it actually occurs via all of the combinatorial effect of all three channels. So I will just quickly go through this, and if you have any questions you can just stop me and ask. The first kind of channel is known as a ATP pump, or sodium potassium ATP pump. So just information, we previously have seen that a cell essentially is a barrier which has a different ionic concentration inside as opposed to outside. So the cell has different concentration of ions inside it as opposed to outside. So there is a lot of sodium ions present outside the cell and a very limited amount present inside the cell, and a lot of potassium ions present inside the cell and few potassium ions present outside the cell. This disparity is essentially brought about by these pumps. They utilize ATP in order to pump in two potassium ions in exchange of pumping out three sodium ions, so that this disparity is maintained. So this is the starting situation. So at the starting situation, the membrane of the neuron stays at the membrane potential of minus 70 millivolt around, and this is constant till any external stimulus is exposed to the neuron. So because, and this membrane potential originates because of the fact that there are many negative ions that are present inside the neuron, and there is a disparity of positive ions in the neurons as well. So we will not be getting into that, but essentially the membrane potential is a negative 70 millivolt, which indicate there are more negative ions inside the neuron as opposed to outside. Once the neuron is electrically excited, what that means is that somehow the electric potential of the membrane, or the membrane potential, increases to some particular value which is known as the threshold voltage. Once this threshold voltage is crossed, the neuron, the other channels start taking over. One of these channels is the voltage gated sodium channel. This voltage gated sodium channel essentially reacts to changes in voltage. So here we have a change in voltage from minus 70 to minus 55, and it immediately opens. It is a fast acting channel. And once you have a gateway that opens directly into the neuron, a lot of sodium ions that were being kept outside start flowing in because of the concentration difference between the sodium ions outside and inside. This immediately raises the membrane potential even higher because positive ions are getting into the cell. So once this one sodium channel opens, another sodium channel immediately next to it will also open, and this will cause sodium channels throughout the neuron to open and increase the membrane potential tremendously, which is known as the depolarization phase. This is how the entire electrical signal is transferred. So this will be as if there is another sodium channel to the left of this image, and we will have a conduction from left to right. So all the sodium channels will open like dominoes from one side of the neuron to the other. This is known as depolarization. And this reaches a peak of around 40 millivolts, which is positive. So the membrane potential is reversed. Once it reaches the peak, another type of channel, which is the slow moving potassium channels, which is also a voltage gated channels, open. Once these potassium channels open, a reverse phenomenon is observed. We know that there are much more of potassium ions inside the cell. All of this start rushing out. So we start losing positive charge, and this losing of positive charge causes the membrane potential to go back to negative. This is known as repolarization, and this is the downward curve we see right here. And it even goes below the original minus 70 because those potassium channels are slow to open or close, and it goes dips just a bit below the normal minus 70, which is known as hyperpolarization, and then comes back to its normal levels. This is how the essentially the entire neuron is repolarized. So it regains its membrane potential of minus 70 millivolts. And but this entire peak essentially occurs throughout the neuron like this, and the signal moves from the dendritic end to the axonal end. So essentially in this image from left to right. So the information, the signal is transferred within the neuron in the form of electrical signals. Any questions from this? Okay. But what happens at the end of the axon? Once the signal reaches the end of the axon, a different kind of activity is observed. The ends of the axons actually connect to the ends of the dendrites of the next neuron. So here we have one axon's end, and here we have the dendrite of the next neuron. This entire thing is known as the synapse, and there is a gap between both these known as the synaptic cleft. Once the electric signal reaches this end, this causes these various vesicles that are present at the end of the axons to uh, fuse to the membrane and still release all of its cargo. What are these vesicles? These vesicles essentially contain chemicals known as neurotransmitters that send the signal from one neuron to the other neuron. These neurotransmitters will go ahead and are released into the synaptic cleft. This will go ahead and bind to receptors on the next neuron. Once they are bound, these receptors essentially are ion channels. These are ligand gated ion channels. These will receive the signal and start opening up. Most of the time these are either sodium or calcium channels, which allow positive ions to go into the next neuron and increase the neuron's membrane potential. If this signal is enough to increase the postsynaptic neurons membrane potential above the threshold potential, then we will see an action potential in the next neuron as well. Essentially the entire information or the signal is transferred chemically to the next neuron. So this is how the synapse works. So coming back to our question, the signal travels electrically within the neuron and chemically at the synapse between two neurons. Any questions from this? So a lot of information about how signals travel between neurons. Just think about it for a second and let me know if you have any questions about this. Okay, I guess there are no questions. So we'll move forward. The next question is, an excitatory signal between two neurons increases the intensity of action potential by tenfold, decreases the intensity of action potential by tenfold, increases the probability of generation of action potential in postsynaptic neuron, decreases the probability of generation of action potential in presynaptic neuron. So four options. You can choose whatever you think is the correct answer and post into the chat. What does an excitatory signal mean? Achal says it is C. Increases the probability of generation of action potential in postsynaptic neuron. Anyone else? Sibani says it is D. Decreases the probability of generation of action potential in presynaptic neuron. Okay, anyone else wants to answer this? Okay, so I am going to talk about excitatory signals and spatiotemporal summation of information in a neural network now. So we have seen that about after our talk with about the synapse, that once a synapse hits a neuron, there are ion channels that open in response. Now, these ion channels that open, depending on what kind of neurotransmitter was released from the presynaptic neuron, these ion channels could either depolarize the postsynaptic neuron, which actually will increase its membrane potential, reaching closer to the threshold potential and allowing for action potential to take place, or it can actually hyperpolarize the neuron, that is, cause more negative charges to accumulate inside the membrane and thus cause it to be even more difficult for an action potential to take place. So depending, if we have a depolarization, we call the neurotransmitter as excitatory, or if we have a hyperpolarization, we call the neurotransmitter as inhibitory. But even if it is an excitatory signal, majority of the time one signal cannot cause an action potential on its own. For example, say there is action potential which reaches the neuron right here. This causes a slight increase in the membrane potential, but this is never close to the threshold, and thus we have no action potential in the next neuron. But what if two of these multiple axons which connect the neuron to the dendrite fire at the same time? So the neurons actually can have multiple axons that connect to different dendrites. Say two of them actually fired at the same time, then we essentially have a bigger depolarization. This bigger depolarization, due to the summation of two axonal firing, can actually cross the threshold and cause action potential to take place. In this situation, the threshold was crossed for long enough that we have two action potentials. And finally, these action potentials triggered uh, action potential in the axon of the postsynaptic terminal. There could be three of the axons that fire together, all of the three connected to the neuron. This causes even higher depolarization, which actually crosses the threshold voltage for even longer, allowing for multiple action potentials to take place, and thus the neuron will send signal, or we call it will fire multiple times. So in this case, where three of these action potentials take place, or the three axons fire at the same time, we have a large depolarization and multiple action potentials taking place. This is known as spatial summation, where different axons fire at the same neuron at different times. It is different neurons acting at the same time. Whereas there is something known as a temporal summation. Say there were three different points at which the action potentials are taking place. This could be due to three different axons or the same axon firing three times. All of them, so once one firing will cause a bit of increase in depolarization, but it is nowhere close to the threshold. The next one will cause even more depolarization, and the third may actually push the membrane potential of the postsynaptic neuron over the threshold and actually cause action potentials. In this case, we do not have, in this case, we have three different time points in which the previous axon fired. This is known as a temporal summation of the signal. So spatial summation, where all the neurons fire at the same time but there are different neurons, so we call it a spatial summation. Whereas temporal summation, it could be the same neuron that is firing at different times, which causes gradual increase in the membrane potential and pushes the membrane potential of the next neuron over the threshold, causing a action potential. So a spatiotemporal summation, which could be very complicated, is what happens in our systems as well when we store memories or decide in different forms of activities such as thinking. So all of these complicated processes occur at the same time to actually allow a neuron to either fire or not fire. So this was about spatiotemporal summation of neuronal signals. So it increases the probability of generation of action potential in the postsynaptic neuron. One excitatory signal is probably not enough to initiate an action potential, but it increases the probability that there will be an action potential. Any questions from this? Okay, moving on. An inhibitory signal between two neurons increases the intensity of action potential by tenfold, decreases the intensity of action potential by tenfold, decreases the probability of generation of action potential in presynaptic neuron, or decreases the probability of generation of action potential in postsynaptic neuron. Keep in mind, this time we are thinking of an inhibitory signal. So you can post whatever you think is the correct answer into the chat. So you can put whatever you think is the correct option in the chat. Achal says it is D. Decreases the probability of generation of action potential in postsynaptic neurons. Anyone else? Anyone else wants to try and answer this? So here you understand that an inhibitory signal will in all probability cause a hyperpolarization of the postsynaptic neuron. So going back to our discussion here, if one of these signals was inhibitory, it will actually decrease the polarization, which will cause it which will make it even harder for the postsynaptic neuron to reach the threshold voltage. So essentially, inhibitory signal will decrease the probability of generation of action potential in a postsynaptic neuron. Okay, next we have a different type of, so we change gears and are asking a different type of question. Efferent neurons, also called motor neurons, are nerve fibers responsible for carrying signals from the blank in order to initiate an action. The options are: efferent neurons carry the information from the brain to the peripheral nervous system, from the peripheral nervous system to the brain, both A and B, none of A and B. You can put whatever you think is the correct option into the chat. Achal says it is A, brain to peripheral nervous system. Anyone else? Anyone else wants to try and answer this? Okay, so this is a simplified image of a stretch reflex arc that we had seen a couple of weeks back. So yeah, so here we have, in case of a stretch reflex arc, we have two different types of nerves that go through the spinal cord. First is a afferent nerve or a sensory nerve. This this nerve, it receives the stimulus from the external environment, could be present on the skin, and conducts the information from the sensory organ right to the spinal cord or the dorsal horn of the spinal cord. Here, this particular information is processed. Either it will be processed in the brain, which will be via the ascending network, and following which there will be information that travels down the descending network, and which will then reach the efferent nerve, which is the motor nerve that will actually do something. This information then travels to the efferent nerve and then conducts the signal to the effector, which could be a muscle or which could be a gland that is then asked to produce hormones or release hormones in response to the initial stimulus. So this is how exactly the entire process works. Sensory organ receives the stimulus, signal is transferred from the afferent nerve, signal is processed either in the spinal cord or the brain, the signal is transferred, processed signal is transferred to the efferent nerve, which travels to the effector, which could be a muscle, and this muscle then contracts in response to the stimulus. So essentially, if we go back to our question, efferent nerves carry signal from the peripheral nervous system, or rather, efferent nerves or motor neurons carry information from the brain to the peripheral nervous system. So the option is A. Any questions from this? Okay. Moving forward, the cell bodies of efferent neurons are present in the central nervous system, the peripheral nervous system, both the CNS and the PNS, neither in the CNS or the PNS. You can put what you think is the correct option into the chat. Dibya says it is A. Efferent neuron cell bodies are present in the central nervous system. Anyone else? Sibani also says it is A. The central nervous system. So going back to our simplified diagram, we can see that the cell bodies of the efferent nerves are present in the central nervous system inside the spinal cord, whereas the cell bodies of the afferent nerves are present outside the central nervous system in the peripheral nervous system in something known as the ganglia. So afferent nerve, the cell body is outside, efferent nerve, the cell body is inside. The cell bodies of efferent neurons are present in the central nervous system. Next question is, cell bodies of afferent neurons are present in the which part of the nervous system? The central nervous system, the peripheral nervous system, both in central nervous system or peripheral nervous system, or neither of those? So you can put what you think is the correct option into the chat. Achal says it is B, peripheral nervous system. Anyone else? Sibani also says it is B, peripheral nervous system. Okay, going to our back to our simplified diagram, we see that the efferent nerve actually consists of, has the cell body, as I said, outside the central nervous system, present in the peripheral nervous system. So the correct option is option B. Okay, so that was all I had for you today. Any questions that anyone has from today's session, then we can deal with those. If no one has any questions, then I will say thank you for joining today's session, and I will see you next week. Thank you everyone.